Well, good afternoon, everyone, and welcome to this session on motor neuron disease and the Brain Conference. Thanks for attending. It's a great pleasure to introduce some fantastic speakers who are at the forefront of our knowledge of ALS, motor neuron disease, and there is a huge amount going on in this disease at the moment, so I hope you'll be stimulated to ask some questions. Please put those in the chat, and hopefully we'll be able to have a good discussion. Our first speaker is Professor Orla Hardiman from Trinity College Dublin. Orla is a major figure in ALS research throughout the world. She's contributed to almost every aspect of the disease, from the genetics through epidemiology, imaging to clinical trials, and she's a great expert in the cognitive aspects of ALS. And so we wanted her to cover really the areas of ALS that may be emerging for people, including cognition, that are non-motor. So now over to Orla's talk.
Good afternoon. My name is Orla Hardiman. I'm Professor of Neurology at Trinity College Dublin, and I'm a consultant neurologist. And I'm going to speak about how as clinicians we approach the non-motor manifestations of ALS. We know that ALS is primarily a motor system degeneration characterised by loss of upper and lower motor neurons. What wasn't recognised up to the past 20 years or so was the other extramotor manifestations of ALS, primarily involving the orbitofrontal circuit, leading to emotional and behavioural dysregulation, the dorsolateral prefrontal circuit, leading to executive impairment, and the cingulate gyrus, leading to motivation. These aspects of ALS are now well recognised and were redefined by a series of consensus meetings which were hosted by Michael Strong and published in the ALS Journal. What was not realised until relatively recently was the more extensive involvement, particularly of language in ALS. So it's associated primarily with cognitive impairment, and we define it as impairment of executive function, which might be multiple domain or single domain, or involvement of either single or multi-domain non-executive impairment, or ALS with FTD. Now, the Ellis-Goyle criteria, which we use to define possible, probable or definite, and then the subsequent criteria that we've used most recently, the Gold Coast criteria, don't require the presence of cognitive behaviour change as a core feature. Some may consider this to be an error, but motor neuron disease, or ALS, is primarily a disorder that's recognised by its upper and lower motor neuron manifestations, notwithstanding these aspects, extramotor aspects, being core for parts of the disease. The ALS-FRS, the functional rating scale that we use, doesn't measure cognition either, so really we're missing an entire domain of the disease at the moment. It's also the case that, as we've taught medical students over the years, that ALS or motor neuron disease is not associated with cognitive impairment, and because the cognitive and behavioural impairment are primarily non-amnestic and may be related to altered behaviour or changes in behaviour, particularly apathy, these may not be recognised by family and may be missed by referring services, and this is often the case in patients who present to our clinic. How do we characterise the cognitive change that occurs in ALS? Well, up to relatively recently, we used very detailed full neuropsychological batteries, which primarily focused on the parts of the disease that, on the parts of the brain that are primarily affected in ALS, in other words, the frontotemporal aspects of the disease, which are characterised by alteration in executive function. There's usually a relative sparing of memory and a relative sparing of visual spatial construct, although language is more impaired than we had recognised heretofore. Of course, this is not practical in a day-to-day setting. Notwithstanding, characterising the nature of the cognitive change is important, and we and others have looked at this over the years. This is one of the first population-based studies of cognitive change, where we can see that up to 50% of patients will have some level of cognitive change, and up to 13% will present with ALS and FTD. What's new about this slide here, which is just published this year, is the presence of language as a significant part of the cognitive change that we see in some patients with ALS. The criteria for diagnosis of ALS with cognitive impairment is well laid out in the Michael Strong paper, which is published in 2017, and as noted, cognitive impairment is divided into those with executive impairment, which is initially frequently presenting with verbal fluency impairment, and then other non-overlapping tests excluding or including social cognition. And these are the batteries that we use, using a full battery lasting approximately 3 hours to define this. And then language impairment, so impairment on some of these neuropsychological tests that we do. So in summary, the most common type of cognitive impairment that we see in ALS is executive function, and that has implications for attention and planning and regulation. And impairment of language, which can occur in the context of word finding difficulties, and also in the more severe forms in difficulties of comprehension. Patients with ALS may present with memory difficulties, which is usually caused by poor encoding and difficulty processing information. So that may be a function really of the executive impairment that we see. What are the clinical correlates of this, and why is it important? Well, the presence of cognitive impairment, particularly executive impairment, is associated with a more rapid disease course. If we look at the slope of the ALS-FRS for those patients who present with frontotemporal dementia established, the slope of decline is almost 4 times that of which we see in patients who have no evidence of cognitive impairment. Those with executive impairment or also have a more rapid rate of decline than those without executive impairment. Executive impairment of itself is a negative prognostic indicator, and different from other types of impairment, particularly language impairment or particularly behavioural impairment. There's a very significant survival disadvantage in those who present with early executive impairment in ALS. And we can see this also if we look at patients who have ALS and look at the various aspects of the disease, for example, bulbar versus spinal onset, the presence of the King's staging system, the years of education. We can see that we can separate patients into 3 clusters, and those with the most severe, least good outcome are those who are most likely to have executive impairment, and those who have the best outcome tend to have relative preservation of cognitive function. Of course, using a 3-hour battery of neuropsychological testing is not really practical on a day-to-day basis. So a number of groups, but particularly the Edinburgh group, have developed a number of instruments that we can use in the clinical setting, and the most widely used now is the Edinburgh Cognitive and Behavioural ALS Screen, or ECAS, which was developed by Sharon Abrahams and Thomas Bach in Edinburgh. And this is what the ECAS looks like. It takes about 15 minutes, and it really is focusing on the aspects of ALS that are most likely to be in the aspects of cognitive impairment that are most likely to be impaired in ALS. In other words, executive impairment and some language impairment. One of the reasons why the ECAS was developed was because many of the existing cognitive screening tools that are available or had been available were confounded by the motor disability that's characterized by ALS. So the ECAS is developed to account for the impairments that we see from a motor system point of view, so we don't get a biased measurement of cognitive change using this screening tool. And this has been translated into many different languages and has been validated repeatedly. The ECAS also has a caregiver behavioural interview, which determines to some extent the level of behavioural impairment that we see in patients with ALS, focusing primarily on the presence of hallucinations, disinhibition, apathy, and loss of empathy. We know that the presence of changes on the ECAS correlates with the clinical progression of disease. In other words, those with the King stage 3 are more likely to have evidence of cognitive impairment than those presenting with King stage 1. This is a cross-sectional study that was published a couple of years ago, but there is a longitudinal study that confirms this finding using ECAS. We can also measure social cognition. That's also often measured using something called reading the mind in the eye. This is work we did a few years ago, showing that those patients with social cognitive impairment also are more likely to have executive impairment. Now, we must be aware that ECAS is a screening tool. It's not a fully fledged developed instrument to fully characterize the presence of cognitive change. It requires training, and it's possible to train an ECAS online by contacting the Edinburgh group and obtaining a certificate demonstrating adequate training. It's also really important in any cultural environment that we have normative values, because even though ECAS is in English and also translation to other languages, it needs to be normalized within that culture, within that environment, within that language structure. It's also the case that an abnormal ECAS should trigger full neuropsychological battery, particularly if there's a low level educational attainment. The ECAS may be erroneous result. This is some evidence that we provided looking at the validation of the ECAS and developing normative data in the Irish population, but we would recommend that be done in all populations for appropriate interpretation of ECAS. What about behaviour? What do we know about behaviour in ALS? Well, as mentioned, there is a behavioural component of the ECAS, but that's really looking at behaviour currently. We developed a behavioural assessment called the Bowman Behavioural Inventory, which looked at the various different aspects of behaviour. There have been variable reports of the type of behaviour that changes in ALS. The vast majority of patients who have ALS have apathy, which is a behavioural impairment, but the behavioural scales really need to correct for physical disability and also for respiratory function. So the Bowman Behavioural Index, or Bowman Behavioural Inventory, looked at all of the aspects that we see, that the aspects of behaviour that we see associated, for example, with the frontotemporal dementias, which is one of the disorders that is linked to ALS. And we developed our inventory using these behavioural impairments that are characterised by FTD. And so one of the important things is determining the presence of new behaviour. Now, this is still a proxy assessment because it requires that the caregiver provides an analysis of the patient's behaviour before disease, and then the analysis of the behaviour during disease. And that, of course, has limitations, because if the caregiver is not aware or doesn't wish to report behaviour, it's not possible to assess that. Now, when we looked at the Bowman Behavioural Inventory and looked at did a factor analysis on this, we could see that there are a number of different subtypes. In our study, about 50% of patients had behavioural change, and we could divide these into apathy, adherence to social norms, social engagement, interpersonal engagement, and self-regulation. And these roughly mapped onto different pathways within the central nervous system, within the insular orbital frontal circuit, the anterior limbic circuit, and the temporal limbic circuit. And this is a measure of the common behavioural changes that we see based on whether there's no behavioural change at all, or whether the person on the Bowman Behavioural Inventory is defined as being mild, moderate, or severe. And you can see that inability to plan is very common in the severe form. Lack of embarrassment, impulsiveness, grammatical mistakes, distractibility, rigidity, perseveration, difficulty control, and aggression. Aggression relatively uncommon in mild behavioural impairment, but inability to plan being relatively common in moderately impaired patients. So to summarise, the most prominent behavioural symptom in ALS is apathy, and there is an apathy scale that's been developed by Sharon Abraham's group as well. That's defined by loss of motivation, initiation, or interest. Disinhibition, the loss of manners, or the new onset of socially inappropriate behaviour. And patients can often present with a reduced awareness of insight or of their deficits and anosognosia. And then there'll be a broad insight a broad lack of insight and loss of social cognition, which can be very distressing for family members. And why it is important? Well, as mentioned, the presence of cognitive change is important because it's a predictor of survival. It's very important that we recognise the presence of cognitive change in terms of decision making, in terms of planning, finances, and end of life decision making. There is some evidence that those with both cognitive and behavioural change may be less compliant with NIV, non-invasive ventilation, and may be less inclined to engage in multidisciplinary care. They may not be as safety aware, so the ability to recognise strategies to avoid falling, for example, or to avoid choking, these can be issues that are very difficult to manage, and particularly in those with behavioural change. But of course, the presence or absence of cognitive and behavioural change is also critical to our understanding of the biology of the disease. It's also the case, particularly, that behavioural rather than cognitive change is associated with a significant increase in caregiver burden. This is work that we've done as well, looking at the factors that lead to caregiver burden, and the patient factors include significant behavioural impairment, apathy and disinhibition particularly, and of course there are caregiver factors as well, including mood and the ability to problem solve pre-morbidly. But the presence of behavioural change is a very significant aspect of caregiver burden. So to summarise, cognitive and behavioural changes are intrinsic aspects of ALS in some patients. 50% of patients do remain cognitively and behaviourally intact during the course of their illness, so that's important to recognise that not everybody with ALS does become cognitively or behaviourally impaired. As the disease progresses, behavioural impairment becomes a little bit more common, particularly apathy. Of those with impairment, early recognition is very important. Screening tools such as the ECAS should be designed to account for the motor disability that we see. They can be very helpful. The ECAS is really standard of care or good practice now, but these tools, the results of these tools, must be interpreted with caution, and the data should be normalised to the population from which the patient has come. If a person has an abnormal an abnormality, or is deemed to be abnormal on the screening tool, it is really preferable that that patient should undergo full neuropsychological testing. And as mentioned, behavioural change correlates with caregiver burden. So what should we recommend in our day-to-day management of those with cognitive behavioural change associated with ALS? Well, first of all, we should recognise that even though cognitive behavioural change doesn't affect everybody, it's intrinsic to the disease. It is part of the disease. It's part of that spectrum between ALS and frontotemporal dementia. It's important that in patients who have early evidence of cognitive change, and that can often be a single domain such as verbal fluency impairment, that we introduce this early in the course of the illness, and that we introduce, for example, the possibility of an enduring power of attorney for a caregiver or another family member, and so that this can be initiated while the patient still has sufficient capacity to understand what's happening and to engage in the process. We should recognise that behavioural change is of greater relevance to caregivers and can be a significant burden, and particularly the behavioural change of rigidity and impulse or impulsivity, and the behavioural change where there's a lack of social cognition, and the behavioural change where patients can sometimes become somewhat manipulative and difficult to engage with, and this leads not only to caregiver burden but also to a withdrawal of engagement by healthcare professionals, or the inability of healthcare professionals to engage from a safety management point of view. We need to recognise that impairment of social cognition is very important. That's often under-recognised, and to reassure families that social cognitive impairment is part of the disease, and that is not that their loved one has not just become selfish and uncaring. That's the disease. We should identify the caregivers who are in need of early targeted intervention, and set realistic expectations for the caregivers, particularly around the behavioural change that we see, and the need to engage with the person, understanding the impact of that behavioural change and how that might impact on the interpersonal social relationships, reduce communication and shared views. And the caregivers should be made aware that the patients may change in their behaviour over time, and that we should probably support and develop the personality characteristics of the caregivers that will help to manage their caregiver burden. So to conclude, cognitive and behavioural change are intrinsic to ALS. Cognitive behavioural change are more likely to occur as the disease progresses. Some patients do remain normal, but if we test people sometimes we do see changes using something called the reliable change index. The patient may continue to score normally, but they may decline from the base from which they have come. Our current testing of cognitive behavior change is semi-quantitative. It's limited to neuropsychological testing, and there are deficits there from that point of view, particularly with respect to behavior change, because we're really using proxy reporting. So there is an urgent need to develop better quantitative measures of cognitive and behavior change, particularly if we're going to incorporate these into clinical trials. So thank you very much for listening to me. This is the research funding that supported the work that we've been doing over the years, and I'm happy to take questions.
Orla, terrific. Thank you so much for packing a huge amount into a short space of time there. Please remind everybody to please put your questions in the chat, but I've got a whole load of questions that I think will keep us happy for the next hour. So firstly, do you think that people with ALS, or at least some of them, have a premorbid personality profile, which may be developmental in origin, that might predict this?
So that's obviously a planted question, Gavin, because absolutely I think that's the case. We've been doing quite a lot of work with first-degree relatives as well, and there's certainly a phenotype, an endophenotype that we see in relatives. So I've come to the conclusion that some of the features of ALS are probably developmental in origin, and particularly some of the cognitive and behavioural changes that we see. Sharon Abrahams and our group who work together, and Carolyn McCutcheon has just completed her PhD, has shown that those who have cognitive and behavioural change are more likely to have family members who have a neuropsychiatric trait as well. So absolutely, I think that's the case, yes.
Yeah, I agree entirely. And so there's a question here. At what stage, from Mark Crook-Rumsey, at what stage along the disease trajectory do the majority of cognitive impairments manifest? Do cognitive behavioural impairments appear before diagnosis, or wait, that we've sort of touched on?
Well, it's a good question. I mean, I think certainly the behavioural change is very difficult to quantify because we're very much reliant on family members. But we do see people who come, whose marriage is broken down a year or two beforehand and who've engaged in very peculiar types of behaviour. With respect to the cognitive changes, usually the first manifestation is an impairment of verbal fluency. And if that occurs, you often will see that progressing over 3 to 6 months so that'd be a single domain form of executive impairment to a multi-domain aspect of the executive impairment. So I think in our experience, when we looked at this in our population-based cohorts and looked at it longitudinally, those who had relative preservation of verbal fluency at the onset tended not to progress, and those who had early evidence of verbal fluency tended to progress. The behavioural change, I think, is a little different. I think we do see increasing behavioural change as time goes on. Behavioural change is not a prognostic indicator, so people with behavioural change tend to survive longer sometimes, or people who survive longer maybe develop more behavioural change. So we could dichotomize out the presence of cognitive executive impairment, which is a negative indicator, and the presence of behavioural change, particularly apathy, and then subsequently other behavioural changes as well, which I think manifests later in the disease, but it doesn't change the survival.
Okay, well, unfortunately, we have to leave it there and move on to the next talk, but thank you so much, Orla, for your wonderful presentation. Got the meeting off to a great start there. So our next talk is from Gavin McCluskey, who's the research fellow in Belfast, and it's about circulating exosomes as biomarkers.
Hello, I'm Gavin McCluskey, and thank you to the Guarantors of Brain for the opportunity to present this work on behalf of my supervisors and co-authors. This is on exploring the diagnostic and prognostic value of circulating extracellular vesicle biomarkers in ALS. My fellowship is funded by the IBN and Guarantors of Brain and an IACN research grant, and I have no conflicts of interest. So extracellular vesicles are small particles released from cells involved in intercellular communication. They're highly stable in the peripheral circulation and can penetrate the blood-brain barrier, and that's made them of interest, particularly in neurodegenerative diseases, and there's been multiple studies over the last several years looking at their role in conditions such as ALS, Alzheimer's disease, and Parkinson's. And Dr. Diaz's team has shown that skeletal muscle in patients with ALS accumulates EV markers compared to Kennedy's disease or healthy controls. And you can see this on the immunohistochemistry slide on the right, which has been stained for CD63, an EV surface marker, and this is much higher in ALS patients. And that led on to this project to look to see if these markers were detectable in serum, and of use as a biomarker in ALS. So for this, we have 75 patients, 30 ALS, 30 Kennedys, and 15 healthy controls, and you can see their details on the right. For the ALS patients, briefly, a third were bulbar onset. The mediated sampling was 64, a mean ALS FRS of 40, and delta ALS FRS of 0.32, so slow progressors. And we isolated the EVs from 200 microliters of serum and then quantified them with a surface marker CD63 and a muscle vesicle marker, as well as their total lipid content. And then on a separate cohort of patients, we performed targeted lipidomic analysis for 15 different sphingomyelins. So initially we confirmed that the particles we identified were extracellular vesicles, and the nanocyte analysis, and you can see this on the top right with a histogram showing the size distribution, and the mean size was 100 nanometer, so appropriate size for EVs. We then compared the CD63 level across groups, and there was no difference in this. But when we looked at the ALS patients, we found that CD63 inversely correlated with age and appears to be higher in younger patients and decline with age. And this wasn't the case for Kennedys or healthy control. Then with the lipid content, again, there was no difference in the total EV lipid between groups, but within the ALS patients, a higher EV lipid content was associated with a higher ALS FRS and a shorter disease duration, so appearing to suggest that it declines with a decline in function and longer disease course. And we were looking at the circulating muscle vesicles. We initially checked that our protein of interest was present on the muscle surface membrane, and you can see this in the immunohistochemistry slide on the left, which is stained green for the protein of interest. And then we were able to detect these in serum via ELISA and found that the muscle vesicle markers were higher in ALS compared to healthy controls, and were also higher than in SBMA, although this wasn't statistically significant, but we are following this up on a larger cohort of patients and also including fast progressors. Then we did targeted lipidomic analysis on 40 patients, 10 ALS, 10 Kennedys, 10 SMA, and 10 healthy controls for 15 different sphingomyelins, and found that 14 of these were significantly different between groups, and 7 of them were raised in all of the motor neuron diseases compared to healthy controls. And one in particular, SMC210, was much higher in the ALS patients compared to any of the other conditions, as you can see in the graph on the right, with p less than 0.0001. So in conclusion, we found that circulating EV biomarkers could be useful for differentiating motor neuron diseases and diagnosing ALS. The EV lipid content correlates with disease severity and duration, and in particular the EV lipid sphingomyelins show promise as diagnostic biomarkers. And we'll be exploring these biomarkers in larger patient cohorts, as well as isolating the muscle EVs by immunosupplementation to further investigate their use in ALS. Thank you very much for your time and for listening to this talk.
Thanks, Gavin. That's really impressive work. I mean, do you envisage these as being a biomarker in a clinical trial? Is that a drug-responsive biomarker?
Well, that's what the hope would be. You know, we're seeming to say that there is some prognostic value with it, and we're looking at the samples longitudinally as well to see if they're declining with time as we think from that initial data. And then it would be useful in a clinical trial to see if it alters the change that we would expect with it. So that's one of the hopes.
I mean, to what extent does this mirror the degree of denervation or muscle burden in terms of weak lower motor neuron weakness?
So we haven't compared it with lower motor neuron weakness. For some of the cohorts, we do have their total examination findings, but the fact that it's going down with time is likely a fact of the progressive neurodegeneration and denervation, which is why we're finding it declining.
Do you have enough subgroup numbers to see whether the bulbar versus spinal onset there was a difference?
In the cohort with 30 patients, we had 10 bulbar and 20 spinal, and there was no difference in that. That's very small patients, so we're going to look at it in greater detail when we do further samples with about a couple of hundred.
Great, many thanks. We look forward to hearing more about this in the future. Thank you very much.
Thanks very much.
So our next presentation is from Professor Jan Veldink from the University of Utrecht. Jan is a major figure in the genetics of amyotrophic lateral sclerosis. He has led the Project MIND consortium, which has really taken this to a whole new level. And I think in his talk, he's going to tell us about some of the very exciting discoveries that are really opening up ALS as a condition of enormous genetic complexity, but this will undoubtedly have major implications for our understanding of mechanisms and for who is at risk of ALS. Now, Jan is actually currently stuck in bad weather in Europe with a poor connection, so he may or may not be able to join us for the Q&A, but we look forward to his talk anyway. So over to Jan.
First of all, I would like to thank the organizing committee for inviting me to tell you about what large-scale genomic studies are telling us about amyotrophic lateral sclerosis, ALS. Well, to start with, why are we doing genetics in ALS? Well, I think it's clear for many clinicians that there's a revolution ongoing within the field of neurology. Really, gene-based therapies are emerging for a number of diseases. It's not always 100% success, but there have been numerous successes already, especially in the field of spinal muscular atrophy, which is spectacular. And I think a good example for especially us in the field of ALS. And so finding genetic variation that underlies disease is a two-edged sword. Of course, it yields concrete targets for new therapies, but it also provides to make true models of disease. You can take stem cells from patients, and these stem cells have a disease mutation, and you can use these models to really screen also for generic compounds and see if they would have any effect in the disease, besides the direct genetic targeting of the defects. So we need genetic targets in ALS. And is there any reason to look for genetic targets? Well, we know that 10% is called familial ALS, meaning there is a first or second degree relative also affected by the disease. But the clear definition of being familial is not there. Lots of confusion, but we do know that for all ALS, DNA is important based on twin studies, for example, but that's not all genetics. There are also non-genetic factors at play. And because of recent technology, whole genome sequencing, and growing groups of consortia, there is this growing list of ALS genes. It's really growing exponentially, as you can see here. But not all these genes are as bona fide an ALS gene as one would like. Really, the top four, the big four, SOD1, TARDP, FUS, and C9orf72 are truly, without a doubt, causative for ALS. And there are some more, of course, but the majority is still unidentified. And all these genes are being curated as we speak. It's a fantastic effort ongoing, the ClinGen ALS GSAP effort led by Matt Harms. And really needed to really say and tell if a variant or a gene is causative, which is crucial for genetic counseling. Okay, so we need genetic targets in ALS. Well, maybe first a small step back. When the human reference genome was completed, with every new DNA sample you would sequence or genotype, you could define genetic variants compared to the reference genome if you are wild type, i.e. identical, or if you're variant to the reference genome. And these variants can be common, and then we call them SNPs. And if they're rare, we call them mutations or rare variants. And but you first have to ask yourself, when you start studying genetics in the diseases, where in this genetic landscape is your disease? So this is maybe the most famous slide in genetics. It shows you the correlation between allele frequency of a variant and the effect it has on humans. For example, on the y-axis, you could call it could be the penetrance, like the chance of getting a disease if you have a variant, or it's the odds ratio, or whatever how you would like to call it. So this is the area of common variants, the area of SNPs that individually have a really small effect, but together can give some higher risk increasing effects for usually common diseases like hypertension or diabetes. And then here in the left upper quadrant, there is the area of Mendelian diseases, rare variants with a huge effect, with this autosomal dominant or recessive disease pattern in pedigrees. And so the question is, where is ALS in this scheme? And so we used to think that ALS, like familial ALS, would be in this left upper quadrant, the Mendelian forms of disease, and then sporadic ALS would be this complex version of the disease. That's important to discriminate because it really governs how you would study these diseases. Since if you have to look at rare variants, you really need to genotype every genetic variant using whole exome or genome sequencing, which is expensive. and it's big data, so it's hard to analyze. Or if you just are interested in SNPs, you can get away with GWAS, with these cheap arrays where these SNPs are pre-plated, so you can simply genotype quickly one sample for maybe 30 euros, and then have your study material. So it's important to know where to go if you can place your disease in this quadrant. And we now know, based on these large-scale genetic studies, we as a community have been doing since 2008, 2009, that this is not the case. This is way too simple to think about ALS, and we do think that the majority of liability to disease is in this intermediate range of genetic variants. So it's not super rare, it's super high penetrance usually, and it's not also the SNPs that are really driving this disease. It's really the intermediate frequency genetic variants that also occur in controls in healthy people or in people with other diseases, but less frequently. And these variants do not have this black and white super large effect, but still considerable effect. And so this actually was the main outcome of our 2016 GWAS, that ALS really is this disease in the middle of these extremes. And so just to jump back to the current day, this is our latest genome-wide association study, which is a follow-up of these previous ones. It includes all the previous studies. This was published in December 2021. And I would like to go through this step by step to see what we have learned from this study. And just to give you a sense of how large scale this is, it's roughly 30K cases and way more controls, because those are usually publicly available, which is convenient. And you can see there is a bias towards European descent samples, which is an issue we need to work on as a community, but this is what it is. And the interesting thing about this study is that we also have whole genome sequencing study samples now based on Project Mine. It's derived from 10,000 samples that have been genome sequenced, and they're also included in this study. And it allows you to really fine map these GWAS signals that we're finding. To cut a long story short, this is what the end result is. You can see this so-called Manhattan plot that shows you all the genetic variants that have been genotyped, SNPs, on all the chromosomes, and on the y-axis you can see the statistical significance to ALS, and you can see it's sort of a Manhattan plot with skyscrapers, which is good. And maybe to remember where we started in 2009, this was the Manhattan plot. It was sort of a Dutch polder plot where there was this tower, this 9P tower, and there was this lonely hit over here. It was kind of underwhelming at the time. You can see this tower really increased in the middle. So that's nice. At the time, it was not known what was driving these signals. And I think that's important to highlight what these kinds of large-scale studies can bring for patients. Because this was a zoomed in picture of that region at the time, 2009. You can again see all the SNPs with their statistical association. You can see there are 3 genes in here, and one of those is C9orf72. But at the time, we didn't know which gene was driving this signal. That's also the limitation of GWAS, that you end up with this signal, but you cannot really fine map or pinpoint exactly where it is. You need other experiments for that. But what it did yield was that at the time it was known that certain families showed linkage to this area, this 9P area, which was way larger than the GWAS region. So this region was sort of fine mapped without us expecting it to only 3 genes. There are 800 genes in this linkage area, so it was fine mapped to 3 genes. And so people started thinking, quite clever, well, let's not look for coding variation in those genes, but let's look at everything in that area, also the intronic non-coding region, which is feasible because there's only 3 genes left. And indeed, 2 years later, so 2011, the repeat expansion in C9orf, the intronic repeat expansion, which is present in maybe 8% of all patients with ALS, also non-familial cases, was discovered, which of course transformed the field. And so this shows you what these large-scale studies can do, especially in ALS, with this architecture where these rare variants with not super large effect, but also not these super small effects are relevant. And as you can see in our latest study, we have this combination of data layers with whole genome sequencing and GWAS And now you can see what happens. This is the GWAS signal as we had in 2009 already, but of course the significance is of course higher. But you can see if we directly genotype repeat expansions in the whole genome sequence, then you can see how fine mapped this signal is. There's nothing in the surrounding, it's really located here in this gene. And if you put these two together in one statistical model, you can correct one out for the other. So they're really, really dependent on each other. It's really nice to see. And so we also pick up other rare variants that are not super Mendelian or super low effect. So this is, for example, the D98 variant in SOD1. And again, you can see that with whole genome sequencing, we find this genetic burden in the whole of SOD1. So all rare variants in SOD1 come up and the surrounding genes are flat. So again, you can see how having both data layers really helps pinpoint these signals. But then we also have these nice combinations of loci where there is a genetic burden of rare variants, for example, in NEC1, which we already knew from other studies. But there are also SNPs, like common variants in the region that are independent from the burden signal. So there's an additional effect of these SNPs on NEC1, maybe by influencing gene expression, which could have a disease modifying effect, which is kind of interesting. And that's not something we see with C9orf of SOD1. And then we have true GWAS hits that truly are common variants where no clear burden signal is residing, or repeat expansions or whatever. For example, in SFCD1, which probably is really a regulatory variant, which has a small increase in risk. So this is sort of the classification we can make based on this latest study. So we either pick up direct coding variants, for example, in SOD1, or pathogenic repeats like in C9orf, but also these genes where both signals reside, for example, NEC1, but also TBK1 is an example, and also these true regulatory variants that we typically see in GWAS, which are complex and usually not too clinically relevant. So these first three are the most clinically relevant regions. But these large-scale studies also tell us more, because you can look at all genetic signals that you pick up, and you can look for patterns in the data and see if other patterns emerge that are relevant for disease. For example, you can look at genes in certain tissues that are highly or lowly expressed in certain tissues and see if your GWAS signals typically co-localize with those tissues. And what we did is we contrasted the ALS signals to Parkinson's, Alzheimer's disease. And what you can see is this distinct pattern of like blood and spleen signals in Alzheimer's disease versus the brain-specific signals in PD and in ALS. And also, if you dig further, you can see that in Alzheimer's disease, you see this microglial effect. And again, which fits the spleen signal, showing that inflammation is really way more important in disease initiation in Alzheimer's disease compared to ALS or Parkinson's disease, which doesn't imply that inflammation is not important in ALS. It might be important for disease progression, but in disease initiation, it seems to be mainly neurons that govern the signal. And as a matter of fact, it's mainly the glutamatergic neurons, which makes sense because riluzole, of course, also the only true disease-slowing drug is targeted at glutamate. And so to further make the point that survival and disease initiation are different, we looked at all these loci from the Manhattan plot, they're plotted here, and see how they relate to survival. So it's a disease progression. In the Project Mind data, we have these data, so in roughly 8,000, 7,000 cases. And as you can see, there's some effect, mainly SOD1, the D98 variant, which is known to occur lead to a slow progression, but C9orf is associated with a faster progression. We know that from the clinic, but the vast majority of these initiation loci, these case control loci, are not at all associated with progression. So this really shows that disease progression is really something else compared to disease initiation. And what you also can do with these data is look at these so-called risk factors that have been studied in the past, physical activity or cardiovascular risk patterns or BMI or all sorts of other risk factors, and use a technique called Mendelian randomization and see if there's proof for a causal role. And we've looked at these these factors, because there's lots of GWAS data out there which you can intersect with the ALS data. And we really only found a causal role for cholesterol, so which is kind of nice to see. And it's not to say that it's probably blood cholesterol, it might be the brain derived cholesterol that's important. So it really shows that there might be a causal role for cholesterol in disease initiation for ALS. And how that translates to treatment, that's still unclear, but it's still an intriguing signal. So what can we target then based on these large-scale studies? Of course, the rare variants, which is true precision medicine. Chris Shaw will tell you later about this. Lots of trials ongoing, exciting developments, but also based on common variants, we do see maybe a role for certain pathways, for example, cholesterol. And we do see a signal in autophagy based on GWAS data. So these might be targetable pathways. And it really leads to the question, is there a role for inflammation, maybe in disease progression versus disease initiation? So what we are planning now is to really, of course, increase case control data sets, but mainly to look at disease progression. We really need this clinical data to get these larger GWAS studies focused on disease progression, because we do expect other, maybe better targets for treatment. And also do this randomization to see if physical activity or cholesterol or cardiovascular traits or BMI, which are so circumstantial or controversial in ALS, if there is some role in causing disease progression. And then my final slides is bigger than always better in ALS genetics? And well, the answer is I don't think so, especially given this specific architecture we've been finding since a few years of these intermediate genetic variants with an intermediate effect. So you can predict that, yes, ALS will tend to occur in families, but maybe not as clear cut as these classical Mendelian ways, but some more distant family members. And so this is sort of the concept here, that if you have a sporadic ALS patient in the clinic with a negative history, family history, but if you would look closely, you could probably find more sporadic ALS patients that in fact are related to each other with maybe 4, 5, 6 generations ago. These people wouldn't know each other. So if you would ask them, they would say, no, we don't know anyone in the family. And so then it would be quite interesting maybe to see if there are other phenotypes in these pedigrees, for example, frontotemporal dementia or some striking low BMI or other related traits we think are related to ALS. And for example, use sophisticated techniques and some, well, clear-cut linkage studies to really find these genetic variants of intermediate effect in these more sort of super pedigrees, as I would call them. So bigger is not always better. This only, well, maybe 30 people, but it would still yield new targets for therapy. So thank you for listening.
So I'm sorry to say that Ian is stranded in a snowstorm and without a connection, so he won't be able to take questions. But I hope that that wonderful talk showed you, particularly those perhaps who are not familiar with the recent advances in ALS genetics, what an incredibly rich biological landscape is emerging. And of course, for patients, the bad news is this is complicated. There's no simple one-stop answer for ALS. But the good news is we are on the beginning of a journey of discovery, which is likely to produce much more meaningful therapies than we've had in the past. So this is all good news in the longer term, and is an incredibly rapidly evolving field. So I would encourage you to look at the recent papers coming out of Project Mine, which I think are absolutely stunning. So our next talk is from Oliver Ziff, who's a research fellow at UCL and the Crick, who is going to talk to us about astrocytes and inflammation, perhaps just as a nice follow-on from some of Jan's comments towards the end of his talk. So over to Oliver.
Thank you to the organisers for inviting me to present this work on the transcriptomic changes in ALS astrocytes. ALS has traditionally been regarded to be due to selecting motor neuron pathology. However, accumulating evidence implicates astrocytes in ALS pathogenesis. I have no conflicts of interest. I searched for all publicly available RNA sequencing datasets of stem cell derived or iPSC derived astrocytes from ALS patients, I found 4 data sets with mutations in VCP, SOD1, C9orf72, and FUS. I first examined each mutation individually and found only a small number of differentially expressed genes for each mutation. But because we were interested to know what changes were common across these mutations and across ALS astrocytes, I pooled them and performed a meta-analysis comparing all ALS astrocytes with healthy controls. There were a substantial number of differentially expressed genes, with 1,300 upregulated, shown in red, and 1,500 downregulated, shown in blue. Gene ontology enrichment analysis showed that genes increased in expression in ALS astrocytes were enriched in ER stress and extracellular matrix, whilst genes decreased in ALS astrocytes were enriched in synaptic and neuronal terms. The glutamate uptake gene set was strongly and significantly decreased in ALS astrocytes. Because of these inflammatory properties of ALS astrocytes, we were interested to know how they compare to neurotoxic astrocytes that have been treated with TNF-alpha, interleukin-1 alpha, and C1q, also known as A1 astrocytes. I searched for all publicly available iPSC-derived astrocytes that had been treated with these pro-inflammatory cytokines and meta-analyzed them in the same way as I did for the ALS astrocytes. I then compared the ALS and A1 astrocyte signatures. Comparing the gene expression changes in ALS and A1 astrocytes, I found a positive correlation, and there was a significant number of overlapping differentially expressed genes. The 381 co-regulated genes were enriched in gene ontology processes, including ER stress and extracellular matrix, and the 497 co-downregulated genes were enriched in neuronal and synaptic terms. This supports the hypothesis that ALS astrocytes acquire neurotoxic features. This schematic of healthy astrocytes in left on the blue and ALS astrocytes in red on the right summarizes our findings. ALS astrocytes display transcriptomic changes consistent with pro-inflammatory cytokine-treated astrocytes with impaired glutamate uptake, ER stress, and immune activation. This work has now been published in Genome Research, where you can find all the detailed results.
Thanks very much, Oliver. So I guess one of the problems in the in vitro context here is that you don't quite know how to map this onto the disease in terms of its natural history. How do you think you can relate what you're seeing in vitro to what's happening in the brain and nervous system?
Yeah, so I guess one of the difficulties here, as you say, is trying to get a model of astrocytes in ALS, and the iPSC approach is one route to do that. In this study, we also looked at animal models, particularly mouse models of ALS, where we found mutations in SOD1 and TDP43, and there was also a membrane model in the mouse as well. So we also looked at those and pulled those in a similar way and found surprisingly very similar findings, that they upregulate these inflammatory genes and they downregulate genes involved with glutamate uptake and synaptic genes. So neither are ideal, but the other model that we could look at is ALS post-mortem tissue. But then the downside of post-mortem tissue is you probably miss that initiation and progression phase, and there's a very weak astrocyte signal in post-mortem tissue. So in this study, we didn't look at post-mortem, but there are others which show consistently upregulated astrocyte, reactive astrocyte processes.
Great. Well, clearly there's a lot more to be learned from this, so we'll look forward to hearing more in the future, and thanks very much indeed for your talk. So we're going to finish this session, I think, on an exciting translational note. Professor Christopher Shaw has been a leader in the genetics of ALS for several decades now, and I think he's always had the ambition that he would turn this into a treatable disease, and I think he's going to tell us about a roadmap for where that might be possible, at least in some of our patients. And motor neuron diseases in general, as has been mentioned already, SMA has been at the forefront of the molecular revolution in precision targeting therapies. So Chris is going to talk to us about that. So Chris is Professor of Neurology and Neurogenetics at King's College London, and we look forward to hearing his talk, Chris.
Good evening. My name is Christopher Shaw. I work at King's College London. and I'm going to talk to you today about targeted gene therapies for amyotrophic lateral sclerosis, also known as motor neuron disease. These are my consultancies for commercial funding. I should also say that I have set up a spin-out company called Aviado Bio with my colleagues Yongbok Lee and Doyong Lee. I need to tell this audience about the nature of ALS, motor neuron disease, as a progressive disorder that starts with disability and gives new disabilities from week to week and month to month. Most patients die due to respiratory failure, on average within 2 years of diagnosis. And it's the most common reason that people seek euthanasia. Perhaps less well known is the disease progression and challenges presented by frontotemporal dementia, which are that patients have a change in personality. They become aggressive, impulsive, often inappropriate. They neglect self-care and generally become very difficult people to live with. And this happens pretty rapidly in the first couple of years, although they live a lot longer than an ALS. These two conditions are connected both in terms of genetic origin and pathology. We know that TDP-43 is the pathology we see in about 95% of patients with ALS, but this is also present in about 50% of patients with FTD. Unfortunately, we can't target TDP-43 itself by knocking it down, even though we do believe it is toxic in some way, and that's because that leads to lethality. Well, these are genes that we know cause ALS and FTD, ALS being the blue and red balls on the time course on the x-axis, and the green balls are FTD genes. Some of them are the same, of course, C9orf72 being a good example, and the size of the balls reflects the frequency of the genetic mutation in these populations. What you can see in familial frontotemporal dementia, at least, we can identify the gene responsible about 50% of patients and about 70% of patients with familial ALS. But both of these sets of genes can be detected in patients with sporadic disease. And indeed, because sporadic disease is more common, the majority of patients with genetic defects will actually have no family history of the disease at all, therefore encouraging people to consider undergoing genetic testing even without, in the absence of a family history. But what can we do with this information? We can start to map out disease pathways that seem to be critical in terms of motor neuron survival and the kind of mutations that occur along the way. We can study this obviously in our patients' brains, but also model it in a variety of cell lines and animal models, work up assays that might tell us about reversing the pathological process with compound screening, taking these back to our animals, and of course then the virtual circles to come back and test these out in man. This would yield therapeutics which might be effective. The disappointing truth is that 20 years or more since the initial diagnosis, 30 years since the discovery of these genes, we really haven't made much progress in terms of developing effective therapies for our patients, which is why I now believe that we should target the genes themselves that are mutated in these individuals to see whether we can make a difference, and also target some genes that are critical to the pathway, common pathway that leads to TDP-43 accumulation. If these are successful, then I believe we should consider screening for patients who carry these gene defects and consider giving them some gene therapy prior to the onset of symptoms. Okay, the most extraordinary results were derived from a clinical trial of an adeno-associated virus given intravenously that carried the survival motor neuron gene in infants with spinal muscular atrophy type 1. Now, these children, the natural history is that they would all be either on ventilators or dead by the age of 2, and none of these children ended up on ventilators, which is pretty impressive. Looking at motor score function, they start off life floppy, and they never sit, they never crawl, they never walk. Many of these children are running around, and some of them have almost completely normal motor function, which is absolutely remarkable. This led to the rapid commercialization of this therapy, which notoriously became the most expensive therapeutic on the planet, but actually has been licensed for type 1 spinal muscular atrophy in the UK under the NHS, but it is about £1 million per patient, but it's a single injection. Well, what's happening on the ALS front? The one therapy that I think we know most about is an antisense targeting SOD1, and this is GAPMA technology developed by Onus and then commercialized by Biogen. and the drug is called tofersen, and they give 100 milligrams, and this data comes from 108 patients. So essentially what we see here is a separation between those patients who are on the active drug, which is the solid line, and those who are on the placebo, which is the dotted line. At this point here, at 28 weeks, all the patients were offered the active drug. Of course, they all went on to the active drug. And so you're looking for a change in the dotted line in terms of the rate of progression. So this is the ALS functional rating scale, kind of questionnaire about people's abilities. And you can see there's very little change in every increase in disability from patients who are on the active drug, and those who were on placebo within a few weeks of starting the active drug were able to stabilize. Unfortunately, those who had rapidly progressive disease made a more disappointing response. Well, one of the ways we measure neuronal death is to look at neurofilament light chain, which is detectable in the cerebrospinal fluid, as is presented here, and in the bloodstream. And what we can see is that there's a very clear separation between those who are on the active drug and those who are on placebo, and that when they all became on the active drug, those lines converge. And then, of course, one of the most important things we can look at is breathing function, and this is slow vital capacity. And again, we're seeing a separation, albeit modest, between those on the active drug and those on placebo initially, and then who go on to the active drug. And again, that tends to stabilize. Sadly, not so much of a dramatic effect for those who have more rapidly progressive disease. So this is clearly promising. We think there is some positive data here. Unfortunately, the trial was reported as showing missing its primary endpoints, which was to show an effect at 28 weeks and good separation. But I think there is going to be important lessons from this going forward, and that SOD1 knockdown is a really important opportunity to challenge this disease. Well, this has been attempted using microRNA, which is a similar sort of process as antisense, but instead of giving it as a chemical injected into the spinal fluid every month, this is given inside a gene that allows the cells to make their own antisense, known as microRNA. And again, targeting SOD1, and this is the work of Christian Mueller and Bob Brown from UMass. And they developed this construct, which they put into a virus, which has got 2 copies of a microRNA, which will bind to and lead to the degradation of SOD1. Well, one of the patients they treated was a very young man, 22 years old, who had what we known to be a very aggressive mutation, A5V, and he had rapid progression in his left leg such that it was completely paralyzed. This is the left leg motor score down the bottom here. But his right leg at the time of therapy, which is day 0, was pretty strong. It wasn't full strength, but it was pretty strong. Unfortunately, in the first 20 weeks, he experienced a radiculopathy. That is, his dorsal root ganglia took up the AAV and elicited an inflammatory response, which certainly must have been miserable for him. And the other miserable thing was that although the right leg strength actually improved and was maintained almost to the end of life, his upper limbs and breathing deteriorated dramatically over this time, and he died of respiratory failure 60 weeks from the onset of therapy. Now that sounds like a terrible result, and indeed it was for that patient, but there was a little bit of a silver lining to the story in that at post-mortem, we discovered that his motor neurons innervating the right leg had been preserved, and that SOD1, in this his case 1 here, was almost undetectable. That means that the vector, this microRNA, was able to knock down SOD1, and that preserved the motor neurons that it got access to. But the problem here is not enough motor neurons receive the therapy and therefore could be saved. Now, a lot of people are looking at AAV to deliver their gene therapies, and most of them are thinking of a CSF route. Indeed, the APIC Bio study I described just before, they injected into the lumbar CSF, whereas many patients now are choosing to inject into the cisterna magna at the base of the skull. Now, of course, that's the outflow of the cerebrospinal fluid, and indeed, CSF is exiting molecules from the brain rather than leading to the entrance of molecules into the brain. We turn over our CSF 3 times a day, and so most of the vector ends up in other organs. The other challenge, of course, is that most people are doing their experiments in mice, which have tiny brains. And of course, vector can perfuse through these brains, sometimes using macaque, and even macaques have got pretty small brains compared to the humans. So the idea that you can get a vector that's sitting outside the brain into the brain substance is pretty unrealistic. And indeed, most of the data in this context shows very small patches of faint transduction, which I think around the Virchow-Robin spaces in non-human primates, even with really massive doses of vector. That's a very large amount of vector. And where the vector ends up actually is in the vital organs. This is a logarithmic scale, and you can see that the heart, lung, liver in particular, and spleen actually receive most of the vector. And when you do cell counting, we realize that less than 1% of neurons are transduced following a cisterna magna injection. And the vast majority of it ends up in organs like the liver and spleen, which is not a good place for these vectors to be. What about the spinal cord? Well, if you were a fan of the lumbar cord, then you'd be reasonably pleased because they get a significant number of motor neurons transduced, up to about 40%. But in the cervical cord, there is almost no transduction of motor neurons. And that's because there's a peel membrane covering the brain and the spinal cord, and a large structure like an AAV vector cannot penetrate through that. It can, however, get into the lumbar cord because there are holes, so-called fenestrations, in the lumbar cord and the lumbar nerve roots, which allow vector to get in and transduce cells. This is not just the work of one lab. There are now 3 labs that reported exactly the same thing. Very little in the cervical cord, and much more in the lumbar cord. So our patients would be delighted to be able to walk around, but they do want to be able to use their arms, hands, and to breathe. So that is not a solution in my book. So what we're going to talk about now is our own work, and it relates to a program we've got on progranulin deficient patients with frontotemporal dementia. We chose progranulin because it's a secreted protein and therefore makes it attractive. And it gives us an idea about how to get these sort of proteins around the brain. We know that the mutations cause loss of function, and that we just want to supplement back to that as a therapeutic. We know a lot about the biology of progranulin, that it's important in lysosomal function in terms of protein clearance, but also in terms of reducing microglial response. In microglia that are deficient in progranulin, they tend to kick out more cytokines and complement, which of course adds fuel to the fire of neurodegeneration. Now, we have pretty good markers of the disease. We know that patients can become, their brains can shrink in size over time. This is serial MRI scans from a patient with progranulin mutation, and you can see that there are just global brain atrophy, but particularly marked in the temporal lobes here. And this happens before people become symptomatic. They're also very powerful biomarker of neurofilament light chain. Again, I mentioned that before, and we see really quite high levels of neurofilament light chain that spike just in the year before people become symptomatic. And there's a high unmet need. We think there are about 130,000 patients in the US and EU that have frontotemporal dementia, and about 5 to 10% of those patients will have progranulin mutations. So that's a real opportunity, substantial population that we really should be looking at to try and develop therapies for. Well, our program, we decided instead of putting vector into the cerebrospinal fluid, which we saw was unlikely to lead to good distribution through the brain, we thought we would use our knowledge of neuroanatomy to target a centralized hub, and that is the thalamus. And these are the different nuclei in the thalamus that project project from the thalamus up and receive input from the cortex. This is the kind of map you get when you look at thalamocortical projections. Well, we use sheep as our model. We are working with Stephen Gill in their unit in Bristol. We do an MRI scan on the sheep, and then we inject into the thalamus. Here is an injection with gadolinium enhancement, and here is an injection just with the vector alone. And these are the kind of results we see. So this is progranulin in the brains of one of our sheep. And you can see this is the chordating the putamen. This is the internal capsule, and you can see pretty good pink transduction. This is the progranulin in pink, and green is the stain for neurons. At higher power, you can see from layers 1 to 6, we get pretty good transduction, almost all the cells. And many of the cells that have got progranulin now are actually glial cells. And that's because the protein has been secreted by neurons and taken up by glia. So this is really fantastic ability to deliver this particular protein throughout the brain. Now, when we compare this with the same vector injected into the cisterna magna, both with AAV9 or AAV1 under different promoters, then you can see there's almost no transduction of the brain in those sheep. But in our sheep, we get fantastic transduction both in the thalamus and the prefrontal cortex, which of course is the target tissue. So just to summarize now, I want to compare the pros and cons of antisense and AAV therapies. Antisense, you know, it's a pretty simple molecule that's able to bind mRNA, lead to the degradation and knock down genes, but it can also be involved in enhancing exon skipping if there's a mutation there that leads to a defect in some way. And there are promising results both in spinal muscular atrophy. In fact, there's a licensed drug for that. And in SOD1 ALS, I think there's a pretty strong hint that there's a positive effect. So it's a powerful proof of concept, relatively low toxicity, not no toxicity, and certainly there is an inflammatory response to antisense oligonucleotides. And there's a relatively low cost of manufacture, although, so lifelong administration in our patients with SOD1, they're receiving that monthly. That makes the pharmaceutical company happy. But there are cons. It's actually a high cost to patients in terms of going in to get your lumbar puncture every month. It's not a comfortable procedure, and there are inflammatory events that happen. And of course, the providers are having to pay quite large sums for these drugs at the moment. It's limited use for loss of function mutations. You really have to find an exon skipping opportunity, which is rare and actually quite difficult to achieve. Of course, antisense will get into every single cell type. So you don't have cell type or tissue specificity. Obviously, you're injecting into the cerebrospinal fluid. Most of it will go actually outside the brain into other organs. And so you could have off-target effects and harm to other organs rather than the brain itself. And of course, a recurrent lumbar puncture is challenging. So lifelong administration of therapy, I say patient unhappy. Well, what about AAV? What are the pros? So you can supplement genes for loss of function. That is reasonably straightforward. Regulating the dose is quite difficult. And there's good evidence from spinal muscular atrophy, and I think some hemophilia that this is useful. There's promising ex vivo, that is taking cells out, stem cells out and putting them back in that have had genetic modification. And indeed, we've seen some promising results from some rare childhood disorders of immune deficiency and in the CNS, adrenoleukodystrophy and hematochromatic leukodystrophy. You can knock down genes using short hairpin RNA and microRNA, and you can undertake gene editing with DNA and RNA using kind of CRISPR-Cas9 technologies. You can, of course, put your gene expression under particular promoters, so it's only expressed in certain cell types, and of course, single-shot therapy, and if it's in neurons, potentially a lifelong effect, which would make our patients very happy. The downside is that there's a very tricky process to manufacture these. There's a very high cost of manufacturing. There's significant risk of toxicity, both with intravenous and intrathecal, because most of the vector gets into other organs. And of course, challenging the dose is difficult, and you can't really stop expression at the moment. And of course, the route of administration can be challenging. In our own route, we're putting this into the thalamus, which of course is a significant advance in terms of invasiveness over a lumbar puncture. Single shot therapy for lifelong effect, perhaps that makes pharma unhappy unless they charge vast sums of money for it. Well, what will really move the field forward, I believe, really advances in terms of the capsid, the protein capsid and the cassette to minimize the dose, perhaps using transient immune suppression to reduce the risk of an inflammatory response. I think improving biodistribution through the brain would be important, enhancing the natural delivery of the vector through the neural network, something we've been looking at, and also regulating gene expression so that if you could switch it on and switch it off or dial up and dial down the dose of the expressed gene, that would be very positive. And then, of course, it's the manufacturing, making stable cell lines so that you can make the same vector in the same way on multiple occasions. And that's very important in terms of regulatory authorities. And then reducing the cost of manufacturing. And we really hope to be able to microdose. Certainly in terms of comparing cisterna magna versus intraparenchymal injection, we're using about 1/1000th of the dose. Okay, so this is the dawn of gene therapies. These are the kind of trials that are underway. SOD1, C9, and ataxin-2. There's also a trial for FUS and C9, and we're involved in all those trials at the moment with patients on those trials, apart from ataxin-2. That's currently not in the UK. And these are the kind of gene therapies that are coming over the horizon. Obviously, Apic Bio has actually treated some patients with SOD1, but these other therapies are in the wings, and I think will be brought through to patients in the very near future. So this is the team that work with me in a variety of capacities. I won't pick out anybody in particular, but just everybody has made a contribution to this work one way or another. And thank you very much for listening to me. These are my typical collaborators, and I have many particular support from multiple sources in terms of supporting this gene therapy work. Thank you very much for listening.
Chris, terrific. Thank you so much. It's incredibly exciting. I mean, we've both been sitting in front of ALS patients for more than 20 years. And it was a big moment for me when we did our first antisense and C9 recently. And it feels like a different era, not without its problems, as you pointed out. So please put your questions in the chat. So progranulin, you're targeting the genetic carriers, I think. Do you think it could be a therapy that might extend out to ALS, sporadic as well as familial?
Yeah, I think we know that it's got a really important role in lysosomal function, and lysosomal dysfunction seems to be a feature of many different genetic forms, but also probably sporadic disease as well. So I think it's worth a shot. At the moment, we're just trying to get it throughout the brain, return it to sort of physiological levels that'll give us some idea about safety. The company is working on this, trying to work up what's an optimal dose, and we have some non-human primate studies underway. But watch this space. I think there are other groups that are taking the intrathecal route, and there are other modalities that are working, an antibody binding sortilin. So there could be some exciting opportunities, and indeed the company that's using the antibody to sortilin has also started a trial in C9 off ALS patients. So we'll find out. It's a biological experiment, experimental medicine. But I think these are really powerful tools, and if we can use them safely and sensibly, we've got real hope of making a difference.
And I mean, presumably you've had conversations with patients about the acceptability of intracerebral injection over other. How do patients get about it?
Yeah, so I mean, if you put brain and needle in the same sentence, most people feel a bit nervous. Of course, functional neurosurgeons, go through the thalamus on a daily basis. It's nothing's like catching a bus. Because they use this in deep brain stimulation. And I think patients will find this more tolerable, particularly if they've had 2 or 3 years of monthly lumbar punctures. They may think, one injection into the brain wouldn't be too bad. I think there is a challenge with conditions like CNN-72 because there's brain and spinal, disease. We have a different strategy for treating the spine, which I can't go into at the moment, but I think we will have to, for C9 in particular, we'll have to develop a dual therapy approach. I think for conditions like SOD1 ALS, we may get away with just a spinal approach because it looks like we can transduce the corticospinal tracts as well.
So questions from the audience. Orla, I wonder whether we know enough to decide who to treat with ASOs and when. The genomic work suggests that risk and propagation are different. Treating after onset might be too late.
Yeah, that's an excellent question. And I'm completely with you on this, Orla. I think that the SOD1 trial shows that most of the aggressive patients got the therapy too late. And we know that SOD1 has got a long half-life. It's about 15 days. And of course, most of the disease is happening down the axon. So if you switch off the signal, there's still plenty of protein being made from the existing RNA and in the periphery. So those are really big challenges. I think the trial so that's my dog. He wants to join the conversation. He's got views, strong views. Yeah, they're doing a study now in which they're taking, I think it's monthly blood tests to look for neurofilament light chain. It might be 3 monthly. And when people spike, I think it's above 19 nanograms per mil, that they will then start their antisense therapy, even though they are asymptomatic. And they will be followed prospectively. We could predict that the majority of those people who do spike with their neurofilaments will go on to develop disease in the next 6 to 12 months. So that will be the test, but it'll be a long and difficult study, and I think powering that study will be challenging. But I've been undertaking predictive gene testing now for more than 20 years, and You know, I've got lots of patients who I think would take this up, and I would love to have a treatment that I really thought was going to knock this down. I think the SOB1 data is still a little bit soft.
Yeah, so we're at the beginning of an exciting journey that has a lot of time to play out, and in particular the bar in terms of safety for treating asymptomatic people is going to be slightly different, isn't it? So that's going to be a challenge as well. We could go on a lot longer, but we've run out of time, I'm afraid. So Chris, thanks so much for your talk, and thank you to all of our speakers for what's been a really wonderful session, and I hope it's convinced the audience that we are in a new era for ALS translational research. So thanks to everybody for participating, and have a great weekend.
Thank you.
Thank you.